The overall goal of this fluorescence in situ hybridization technique is to concisely analyze telomere number and length in C. elegans.
This method can help answer key questions about tumorigenesis, alternative lengthening of telomeres, and cellular senescence.
The main advantage of this technique is that the procedures are concise and the telomeres can be visualized and quantified in less than 24 hours.
Collect the gravid adult worms with a little liquid media, without debris, in a 15 milliliter tube. Now, wash the worms 3 times as follows. Add M9 buffer to a total volume of 15 milliliters. Then, spin the tube down at 300 G for 3 minutes and discard the solution above the pellet of worms. Then, repeat the process. If after the centrifugation the worms are floating, then lighten the brake on the centrifuge. After the third wash, leave the worms with 0.5 milliliters of M9. Then, add 6.5 milliliters of distilled water, 2 milliliters of hyperchlorite, and 1 milliliter of 5 molar potassium hydroxide. Let the worms incubate in this bleaching solution for 3 minutes with 50 RPM of agitation. Then, vortex the worms to shear them open and free their eggs. Under a dissection microscope, look for the released eggs. If they are not released yet,